Why won't these cells die? Scientists are testing whether aging itself can be rewritten. One of the first clues came from a cell that should have quietly disappeared, but didn't. Instead, it lingered, slowly damaging the body it was meant to protect. This isn't a story about cancer. It's about a biological defense mechanism that works for decades before becoming one of the forces driving aging. It's called a senescent cell, and by the end of this video, you'll understand why it may have changed aging research forever. To understand a senescent cell, you first need to understand what it was meant to do. Every cell carries a built-in timer. Each time it divides, its telomeres, the protective caps on chromosomes, become shorter. After enough divisions, the cell receives a simple message: stop. It doesn't die. It remains alive but stops dividing. This is called cellular senescence. For decades, scientists believed this was a powerful defense against cancer by preventing damaged cells from multiplying. They were right, but only partly, because these cells don't always disappear. Many remain in the body, slowly changing the tissue around them. And that discovery would completely change how scientists understand aging. Here's what scientists eventually discovered. A senescent cell doesn't simply stop dividing, it starts sending signals. It releases a mix of inflammatory molecules, enzymes, and growth factors known as the senescence-associated secretory phenotype, or SASP. Think of it as a distress signal, calling the immune system to remove the damaged cell. In a young body, that's exactly what happens. The immune system clears the cell, the tissue recovers, and the process ends. The problem is that the immune system ages too. As we grow older, it becomes less effective at removing senescent cells. They begin to accumulate, and the signals that once called for help never stop. Instead of protecting the body, they create a constant state of low-grade inflammation that slowly damages the surrounding tissue. Over years, this silent process contributes to stiff joints, slower healing, declining brain function, and many other hallmarks of aging. This isn't random wear and tear, it's a biological mechanism. And that realization changed everything. Senescent cells aren't just a consequence of aging, they're one of the processes actively driving it. In 2011, researchers at the Mayo Clinic set out to test a simple but radical idea. What would happen if senescent cells were removed instead of allowed to accumulate? Led by Darren Baker, the team genetically engineered mice so that only senescent cells could be selectively eliminated. At the flip of a genetic switch, the so-called zombie cells died, while healthy cells remained untouched. The results became a landmark in aging research. The mice lived longer, but more importantly, they lived healthier. They developed cataracts later, maintained stronger muscles, healthier hearts, and better tissue function. Instead of spending their final years in steady decline, they remained physically capable for much longer. Then the researchers asked an even tougher question. What if the damage had already begun? They waited until the mice were old, showing the expected signs of aging, and only then removed the senescent cells. It still worked. The mice didn't become young again, and existing damage wasn't erased. But tissue function improved, physical decline slowed, and several age-related problems became less severe. Removing the source of ongoing inflammation changed what happened next. The implication was profound. Senescent cells weren't simply a consequence of aging. They were actively driving it. And if removing them could improve health even later in life, aging might be far more treatable than scientists had ever imagined. The Mayo Clinic findings sparked an entirely new field of research. If removing senescent cells improved health in mice, could the same be done in humans? That question led to the development of senolytics, drugs designed to selectively eliminate senescent cells while leaving healthy ones untouched. Early human studies have produced encouraging results. Compounds such as dasatinib and quercetin have reduced senescent cell burden in human tissue, while companies like Unity Biotechnology have begun testing similar approaches for age-related diseases. These are not proven anti-aging treatments, and they are not available as routine therapies. Human trials are still in their early stages, and many questions remain. But what has changed is the pace of progress. Just a few years ago, senolytics were a niche idea. Today, they are being studied by hundreds of researchers worldwide. That shift reflects growing confidence that senescent cells are more than a symptom of aging. They are a real biological target, and that scientists are now learning how to control them. Now for the reality. Senescent cells are one driver of aging, but they are not the whole story. Removing them in mice significantly improved health and extended healthy lifespan. Yet the animals still grew old and eventually died. Aging is driven by multiple biological processes that interact with one another. Solving one does not stop the others. But that's exactly what makes this discovery so important. It shows that aging isn't a random accumulation of damage. It's a collection of identifiable mechanisms that scientists can study, test, and potentially influence. 20 years ago, that idea seemed out of reach. Today, it's reshaping the field. Aging may not have a single cure, but if each of its underlying mechanisms can be understood and targeted, then slowing or even partially reversing aging becomes a scientific challenge rather than an impossible dream. We now know aging isn't random. Senescent cells showed that the body follows biological mechanisms and that some of those mechanisms can be changed. They weren't the whole answer, but they proved we were asking the right question. The next mechanism is even stranger. It's not about what cells do wrong, but what they forget, and whether those forgotten instructions can be remembered.